The DNA fragments separated on an agarose gel can be visualized after staining with which dye? Option A, acetocarbene. This is incorrect because acetocarbene is a nuclear stain and it is usually used to observe chromosomes under microscope. But it is not suitable for staining DNA fragments in an agarose gel. Option B, aniline blue. It is also incorrect because aniline blue is mainly used for staining cell walls and it does not bind to DNA for visualization in gel electrophoresis. Option C, ethidium bromide. Now, ethidium bromide intercalates between the bases of the DNA and it fluoresces under UV light. So, it is an ideal stain for visualizing DNA fragments in agarose gel. Hence, it is the correct option. Option D, bromophenol blue. It is incorrect because bromophenol blue is a tracking dye that is used to monitor the progress of electrophoresis and it is not used for staining DNA.